Hi, my name's Anna Padavan. I'm filling in for Professor Karen Gibb, who couldn't be here today. Karen leads the Environmental Chemistry and Microbiology unit. This group does research and collaboration across all of Northern Australia. We specialise in high-end trace element analysis, stable isotope analysis, nutrients and also microbiology analyses. We don't just produce raw data, but we like to tailor our research to the specific needs of research and clients as well. The group has about a dozen people. We have research assistants, TOs, research fellows, 4 PhD students and a master's student, and as well we have admin support with an admin leader to deal with the budget and other issues. And we're located in Yellow 2. Some of the projects or larger programs that we're involved with are divided roughly into 4 themes. We are involved with healthy marine harvest. This is looking at anthropogenic impacts on shellfish in Darwin Harbour. And in this work we looked at trace elements and microbial communities in shellfish. And down the track we would like to fine-tune that and rather look at whole communities, have a bit more of a focus on individual or groups of bacteria, such as the E. coli human faecal markers. A second area is in marine health and baseline assessment. In this part of the research, we do trace element and stable isotope analysis of sediment and biota. And this is an area that Kanchana's PhD project falls into, and she'll describe some of her interesting work from Buffalo Creek in a minute. The third area is the microbiology of waste ponds and tidal creeks. And in this area, research is focused on microbial source tracking, bacterial diversity, and also bacterial function, especially in relation to nitrogen cycling. And some of the results from this work are already leading to monitoring of faecal indicators and pathogens in Darwin Harbour using molecular tools, and also looking at the function of the ecosystem using these nitrogen cycling genes. And Miriam is a research fellow on an ARC linkage project working in this area, and she'll have a few slides at the end to highlight some of this work. And finally, the group has a mobile isotope monitoring unit, and it's been used to look at water dynamics and water budgets from creeks and rivers through to oceans. And it will be used also to look at carbon and nitrogen cycles. And we've already heard a little bit about that in the talks. And Lindsay's group is embarking on this, or using this technology as well. So to some research highlights in these 3 areas, this plot is a summary of microbial community analysis in shellfish and sediment and water from Darwin Harbour. So each of those dots represents a bacterial community, and each of those dots represent hundreds to thousands of different bacteria. And so what we can see from this is that microbial communities in water and sediment are quite unique. They're different to each other and different to the shellfish. Oysters also have their own unique microbial makeup, and even though they sit in the water and filter water, they have a different microbial community than what's present in the water column around them. And similarly for 2 other shellfish species, mangrove snails that live on stems and roots of mangroves, and also mudwhelks and longbums that live and feed in mangrove sediment, also have their own distinct microbial populations. So the bacterial communities that we're seeing in the biota isn't the same as what's in the sediment and the water. And what we hope to do with this work is take a closer look at each of these groups individually to see whether there are bacteria or groups of bacteria which are specific to different areas of impact in Darwin Harbour. And by doing so, try and understand what drives the selection of bacteria and what the implications might be for monitoring. So I'll let Kanchana talk about her isotope work.
Thank you, Anna. My study aimed to look at the impact of treated sewage discharge on Buffalo Creek ecosystem using stable isotopes. Stable isotopes are chemical traces that naturally exist in the environment. So why are they important? Because isotopes in sewage effluent discharges have higher values than the values that are recorded, background values recorded from unimpacted ecosystems. And also, these high isotope values will pass into primary producers like mangrove trees or consumers like snails when they feed on wastewater-derived nutrients. So, measuring isotopes in biota will help to determine the spatial extent of sewage plume and to which degree it is incorporated into biota. This slide shows some interesting results from my project from snails in Buffalo Creek. Both snail species sampled in Buffalo Creek showed high values compared to the snails sampled from the reference creek. The highest values were recorded at the treated sewage outfall site, and the values gradually decreased along the creek, and at the creek mouth, that's where the boat ramp is, the values reached the ones similar to the ones that were recorded in the unimpacted creek. The mangrove leaves also showed a similar trend. These results suggest there is a substantial uptake of wastewater-derived nutrients by mangroves and consumers along the entire length of the Buffalo Creek. The outcome of my study will assist in developing isotope tools for long-term monitoring of this ecosystem and also will help to understand the effectiveness of the current sewage treatment upgrades and will allow to take suitable management actions. Thank you.
So the next slide is a summary of work done by another research fellow, Niels Munksgaard, and it was a sediment core study done in Rapid Creek. And the aim of the study was to look at the history of pollutants in sediment in this area prior to starting a sediment monitoring program. So in this study, 4 sediment cores were taken along Rapid Creek, 3 along the creek itself. Can you see the mouse there? Yeah. And 1 in the floodplain. And these cores were analysed for metals, for lead isotopes, and also for dating. So what one of the cores, the RC1 core, showed that was very interesting. It was the only core that could be dated, and the results from this core are shown in this blue line. So you can see that copper concentrations in the deeper and older parts of this core were actually quite low, up until about 14 centimetres, where there was an increase in copper, and that level remained high. This 14 centimetre mark in the core corresponds to about somewhere between 1923 and 1944, so loosely around the time of, or just after World War II, which is when, or after which Darwin started to develop quite rapidly. And a similar trend could be seen with the lead isotope ratio here, where the ratio was quite low up until, again, that World War II or development period, where the isotope ratio increased. And this can be correlated to the use of leaded fuel. So these cores provide like a bit of a historical story on pollutant loads that were there beforehand. What is good is that these copper concentrations and lead isotope ratios in other metals that were also measured have remained stable since development, and they're still below the ecosystem guidelines. The other core samples, these two here, the green and the red, were too mixed for any dating to be done, but they show the higher amounts of copper concentration and also the higher lead isotope ratios. So what this says is that the sediment present in these core samples were all deposited after World War II, so post-development. The final core sample, which was taken from the floodplain just here behind the university, has low concentrations of copper and low lead isotope ratios. So it hasn't been exposed to any new sediment and shows that there's very infrequent flooding and infrequent deposition of mangroves in that floodplain area. And Miriam will finish up with her nitrogen work.
Okay, so within an ARC linkage project with partners, Impacts, Northern Territory Government and Power and Water Corporation, we're looking at the impact of treated sewage effluent upon the microbial community in Darwin Harbour. So over 2 years, we collected sediment and water samples from 2 effluent impacted creeks, the Buffalo and the Myrmidon Creek, and 4 reference creeks. And amongst other things, we looked at how the bacterial communities change with increasing distance from the outfall of the wastewater stabilization ponds, such as this one at Buffalo Creek. And we saw a surprising match between the actual distance from the outfall and our sampling sites and the relatedness of the bacterial communities, which we assessed by 16S DNA next-gen sequencing. And we can see here that the bacterial communities slowly reverted back to those seen in reference creeks in Jowl Bay once they were past the boat ramp. But with sewage effluents having such a large impact, obviously, on the nutrients in the water, they also have a large impact on the nitrogen cycle. And so we also looked at nitrogen cycle function and markers. And we used this novel functional gene array, which was developed by CSIRO Hobart, which targets the entire nitrogen cycle. And we do this to see whether changes in the nutrient levels in these water samples are reflected in changes in gene abundance of microbes, which are associated with the nitrogen cycles. And we got some promising initial results, such as from Buffalo Creek outfall, where we saw very few gene signals from 4-nitrogen fixation, and signals for denitrification were strongly reduced as well, which nicely matched the nutrient data, as well as for a Myrmidon Creek outfall, we saw strongly reduced ammonia oxidizing thickness, which again matched the nutrient data where we had very high ammonia levels. So all up, these are very promising initial results that this functional gene array will be or is a good tool to measure ecosystem function. And we are now looking into developing biological indicators to measure ecosystem changes in ecosystem health based on this array.